Hello everybody, today we gonna talk about the gene ontology which can be done by a panther tool. It is basically an classification tool which provides us classification of our data. So basically I have a transcriptomic data. Here is it. So first of all, what I'm gonna do that I'm copying the specific IDs of my transcriptomic data. If you have any kind of FASTA data, that can be RNA sequence data or cDNA or rod sequence data, different kind of data. So you just have to copy your accession number and then paste it into another section. So here I have already done it because it's time consuming. So first of all, we must copy that section and paste into go to the new tab and enter the panther tool pathway analysis. Here we have, so it's in time consuming because you will be uploading a large number of data. So first of all, upload your data. I just pasting my data by pressing Ctrl and V. Okay, so we have to select. You can also select a file, like for example you can choose this file simply. So I just copy my data manually. Then we move to the next section. So we can also explore our work again, which we have already done previously, but we will do it from our ID list because we are doing for the first time. So we will be selecting our species. So for example, my species Arabidopsis theriana. Okay, then move to the third part. So here we have, you can also retrieve a function classification of genes, like they will give you some sort of list, and also you will have some graphic charts like this. So whenever you submit this, so it will take a little bit time because it's time consuming. We have uploaded a large number of data. So first of all, these sequences must be explored by this side, and then they will classify it. So I have already done this before to submit that. So this will give us a result like that. Okay, so these are a kind of our result from our data. So they give us a full explanation regarding our data, that how much our data contains, like in molecular function, here in biological function, cellular component and protein class. So let's move to the biological process. Okay, so here is it, the result. They give us a full explanation of our data, that how much your data contain of these things. Like they give us explanation like these data cannot be assigned to the panther because these data cannot be classified or accepted by the panther database. So we must remove these data because it's in kind of raw data. Okay, so here these give us a full explanation. Like for example, the list we have uploaded, so in that list 938 was in like genes which involve in localization as well as cellular process. There were total 4645 genes, as well as they give us a percentage according to the chart and according to our data. Like for example, the 33, if you look at to that, so it can't be shown here. So anyhow, so they give us explanation like 32 is the coverage according to the chart table, while 22% is the coverage regarding to our data. Okay, so what we will gonna do, we can also download these, but that will be a bad result. So first of all, we must export this data to the chart, sorry, and then copy these data. Okay, and now we are paste it in a section. So they give us a full explanation. So here is it. In cellular process there are total these gene involved, like these, like these, like these. So we must create a chart, which we will do it now. So sorry, I'm creating in PowerPoint because whenever you create some sort of chart, so that will be based in PowerPoint. Okay, so we must move to the chart, pie chart. Now what we will gonna do, just paste our data. Sorry, once again we will try. We will copy the same data because we have don't need these data, so we will simply copy these data and paste it to the another section. So here is it, our data. Now to provide us data full explanation, we must give it a better look to our data. So it's kind looking awkward. Okay, so first of all, what we will gonna do, that make it a best fit data. Okay, so then we will to make sure that all of the data uh, can be expressed so the reader can understand it easily. So now we are adding it in biological process. Okay, same as well as we can download all of the data as I downloaded in molecular function and cellular component as well as in pathway. So they gave us because our genes, we can say that these were a transcriptomic data. So in transcriptomic data, that cannot be enriched with the pathway database, so that's the reason they gave us a bad result. You see, here we have almost 88% of the data which is unclassified by the panther database. So basically it isn't a pathway database. For pathway we use KIG, metasec, reactome and other databases. Okay, so these are kind of just classifications. So that was a review about the panther database, and that is the latest version. So this was a kind of gene ontology, so we have done it. If you like this video, so kindly like my video, share it and subscribe my channel. Thank you.